So I think this YouTube stream is set up. And in 1 second, I'll open the waiting room if everyone's ready and we can get started. You guys are all okay. Let me do that then. All right, so thank you everybody for joining us for another session of the PC3 seminar. And if you guys don't know, PC3 stands for Prebiotic Chemistry and Early Earth Environments. And so we really want to try to bring together some of the different researchers in these different fields. And before we get started today, we have a quick announcement about an upcoming PC3 workshop that'll be happening in a couple of weeks. So I think we have Zach Adam here to make the quick announcement. So if you're
wonderful, thanks, James. You should be able to see the announcement on your screen now, and if you don't, you can find it on the PC3 website itself, or the RCN. The workshop is really going to focus down on system studies. So a lot of PC3 research is about into a particular compound or a particular setting or a particular pathway or some kind of biological portion. But when you go from individual components up to a system level behavior, it's a jump in magnitude and complexity in terms of experiment design, in terms of the way in which we approach the study of those systems. And the workshop that's coming up in October is just going to be about evaluating complexity at the systemic level, both for geochemistry, for cosmochemistry, for organic chemistry. And so we've got 4 different sessions. The sessions with formal scheduled speakers will take place on day 1 and day 2, and day 3 will be dedicated almost predominantly to discussion and synthesis of how we try to make sense of these systems and how we might best design experiments to study things at the systemic level, or to best try to replicate some kind of interesting systemic behavior. So again, you can find this information on the PC3 RCN website, and register for the register for the workshop ahead of time, because it will be a controlled access kind of workshop. We'll all be working together with shared documents and so on. So I look forward to as many people as possible attending, and I hope to see you in about 6 weeks. Thank you.
great. Yeah, that sounds like it'll be a great time. So thanks for bringing that to our attention, Zach. And so now we'll, I guess, just hop into today's session, which is going to be, theme for today is biosignatures. And so we'll be hearing from, we'll have a quick introduction from Don Sumner, and then we'll be hearing from Erica Barlow and Josh Krissansen-Totton. And before we do that, we've been just kind of running this quick poll just to kind of figure out who is really showing up to these seminars. So I'll put that up really quickly, and you can kind of take that as I perhaps introduce Dawn and she gets set up. So that should have popped up on your screen. And so yeah, so first we'll be hearing from Dawn Sumner, and she's a professor at the University of California, Davis, and she received her PhD from the Massachusetts Institute of Technology. And her research focuses on early Earth environments and microbial ecology, emphasizing evolution of oxygenic photosynthesis and its environmental effects. Dawn also studies microbial ecology in ice-covered Antarctic lakes as analogs for ancient life on Earth and possibly on Mars, using data from NASA's Curiosity rover. And Dawn is also dedicated to curtailing racism and promoting inclusive practices in academic settings. So thank you for joining us, Dawn, and go ahead.
There we go. It's really a pleasure to be able to introduce the topic of how we look for life, evidence of life on early Earth, and how we look for evidence of life on other planets. I really enjoy the image I'm sharing with you that's taken under in an ice-covered lake in Antarctica, because it looks like an otherworldly view. Microbial communities here are composed of photosynthetic organisms, and their whole complex community that makes an ecosystem. But it seems really unfamiliar to us because we're used to looking at other more broadly distributed environments. But one of the other interesting things about this is that there are fossilized microbial communities on early Earth, particularly in the Archean, that look like this, but these particular ones aren't preserved because they're composed dominantly of organic matter with no mineralization. And so we've had life on Earth abundantly, probably on essentially every environment on the surface of Earth for billions of years, but our record of that is difficult to parse out. On the modern Earth, sort of every part of the surface and atmosphere is influenced by life. And this is one of the things that I think makes it hard for us to think about looking for life on other planets. So for example, Mars, where if there was ever life on Mars, it's very sparse. And this gets at this idea of our planet having a biosphere that really dominates the whole planet, leading to ideas like the Gaia hypothesis, where life acts as a whole system with its planet. But we are an exception for at least the planets within our solar system. And if we go back to early evidence of life on Earth and the fossil record, which Erica is going to talk about, the rocks like this one that formed, this is a 2.6 billion year old rock. We know that microbial communities were covering most surfaces. This one has an appearance of stromatolites vaguely, but it's actually formed from mineral growth processes. They're carbonate minerals, which are often influenced by life, but it's really difficult to prove that these particular ones were influenced by life. On Earth, we know much more about the history. When we go to another planet, like early Mars, so this is in Gale Crater from the Mars Curiosity rover. The white line near the bottom is a sulfate-filled vein it's about 3/4 of a
Sorry,
centimeter.
at least for me, the slides aren't advancing. Not sure if that's just me or if anyone else is having the same problem.
It should be a reddish slide.
Yeah, it looks like we're still on your initial slide. I'm not.
Oh dear, okay. My initial slide, that is really, so do you see the non-presentation mode now?
Yes, yeah.
Okay. Did the slide change now?
Yeah.
Okay, I don't know why it wasn't showing in presentation mode. This is one I was talking about. Maybe I'll just do it.
That looks fine.
Okay, we'll just do it this way.
Yeah, thanks.
I have no idea why. Anyway, I've been doing this for years, and this is the first time that's happened. Okay. Well, you got to look at Lake Vanda for a long time. Right. So this particular one is it's a photo looking down on a bedding plane of Mudstone on Mars. The mudstones that we've analyzed have organic matter in them. They have energy sources in addition to possible photosynthesis, but it's very difficult to actually find a signature that can tell us whether or not those organic molecules came from life. Luckily, the Perseverance rover is bringing samples back that will help with that. And so when we think about detecting life on other planets, on Earth, the atmosphere is highly influenced by life, and we are sending out cell phone signals, and SETI is sending out electromagnetic signals that someone else could detect, and looking for the same from other planets. But the types of things we can look for are on extrasolar planets as possible biosignatures are really challenging. But it's one of the most exciting types of science to work for, asking what is the context of life on Earth in the context of the universe, and is there life elsewhere? And so Josh and Erica are some of the early career people who are working on some of these problems, and I'm super happy to turn it over to them to talk about their work.
Great. Thank you so much, Don. And now we're going to be moving on to hear from Erica Barlow, who's currently a NASA postdoctoral program fellow working with Chris House at Penn State. And she earned her PhD in geology at the University of New South Wales, Sydney, where her research focused on morphological, textural, and isotopic biosignatures in the early Earth rock record. And her current research with the Laboratory for Agnostic Biosignatures explores the idea of universal signs of life in rocks to contribute to the development of tools to be able to recognize and describe fossilized life as we don't know it elsewhere. So thanks for joining us, Erica, and go ahead.
Thanks a lot, James. Can I just confirm you can see the proper view?
Yeah, that looks good.
Great. So firstly, thank you for the organisers for inviting me to give a talk on my research today. James, Becca, Danielle, Albert, I'm sure there are others as well behind the scenes. So as James mentioned, I'm a NASA postdoc at Penn State, and I work with the Laboratory for Agnostic Biosignatures. And my background is looking at biosignatures in the early Earth in rocks. And so I'm going to share some of my research today about what that's all about. So just okay, sorry, it wasn't advancing for a second. Just to give a quick overview of where I'm going so you can follow the talk. I know we've got a diverse audience here. I'm going to give an introduction into what biosignatures mean in my context. I'm going to talk about assessing the timing and origin of biosignatures, the terms that we called syngeneity and biogenicity, and then move on to some examples of what can signs of life in rocks actually tell us about the life and the environments that they formed in, and then provide two case studies. The first one on some information from Turi Creek group rocks, which are about 2.4 billion years old. So that's where I did my PhD work. And then some information about where I'm currently heading with agnostic biosignatures and how that all ties in. So to begin with, types of biosignatures in my context, where I come from. So there's two broad categories, morphological and geochemical. When we're looking at the morphological biosignatures, these can be both macro and microscopic, and they can also include textures. So it doesn't just have to be a body fossil or a macro fossil like a stromatolite structure. It can be textural as well. So here we have an example of a macro fossil, a stromatolite or stromatolites from the 2.7 billion year old Tumbiana Formation in Western Australia. Then we also have an example of microfossils. They're individual filamentous, likely bacteria with potentially cells preserved. You can kind of see a light and dark striation, at least the hints and the remnants of the cells there. And these are 2.4 billion years old from the Turi Creek group, some of the work from my PhD. And the microtexture example I'm showing here is of what's inferred to be bubbles preserved from the 3.5 billion year old Dresser Formation in Western Australia. And more about those later. So the other main category of biosignatures are geochemical. So these could include elements, organics, minerals, and isotopes, just as an example of the categories. So when we look at the elements here in the center, the trio of images, this is from a really neat study by Sumner et al. In 2014, showing the same image three different times, highlighting the different elemental distribution. So in the top, there's calcium, which really highlights the little stromatolites and the layers and the bumpiness, the morphology. Then when we go down to the iron and the arsenic, we can see that these elements are actually co-located within the stromatolites. And so these authors, along with other geochemical evidence, inferred that this could be indicative of arsenic cycling. And these are from the Tumbiana Formation at 2.7 billion years old. There's also organic biomolecular studies. This particular one is from a study by Brox et al. On 1.64 billion year old rocks from the Barney Creek Formation. And we can also look at minerals and isotopes. They give us a wealth of information. And here the mineral is actually pyrite, and the color change is showing variation in the isotopic composition of sulfur. So you can co-locate these different types of biosignatures. And actually it's often much better to do that if you can combine multiple types of biosignatures, because it gives us much more information when we do that. And going back further in time, it's even more important to do this because there's not as much information preserved to begin with. It becomes a little bit messy because there's diagenesis and metamorphism, so post-depositionary processes that alter what you're looking at. So as we go back further in time, you get much better picture when you combine multiple types of biosignatures. So now just briefly about how we assess if a potential biosignature is a biosignature. So the two terms I'm going to refer to are syngeneity and biogenicity. So syngeneity refers to the age or the timing of the structure in question in relation to the surrounding material. So I've got an example here of a stromatolite, just as a morphological biosignature. It's easier to kind of get it, you see what I'm getting at, but the stromatolites are outlined here in blue, and the surrounding sediment is shown here in the orange. And you can see that there's this distinct relationship where the stromatolite existed and the sediment unlapped up against it and filled in between these structures. So we know that this stromatolite formed at the same time or in the same sequence of events as the sediment. It's not coming from some later input or other event. So this is confirmed by what we see in the modern day, where stromatolites stick up above the sea floor and the sediment builds up and fills in alongside them. So this is how we know that they're syngeneic. Another example, using morphology again for biogenicity. Biogenicity refers to the origin of what you're looking at. So is the feature of interest actually biological, or could it have been produced by an abiological process or processes that mimic biology? So an example here is stromatolites again on the left, and this is a neat outcrop. This is from South Africa, and you can see the stromatolites both in plan view and in cross section, so you can get a sense of the 3D morphology and what they look like from the different angles. And an abiotic mimic, at least on a cursory kind of comparison, could be something like mud cracks that make these similar polygonal shapes on the surface. But a way to distinguish between them is that mud cracks don't penetrate very far down. So if you get a sense of the 3D shape of the structure you're looking at, that's one way to tell or to distinguish between abiogenic and biogenic structures. So whenever you're looking at anything from the early Earth, I mean, obviously Mars and elsewhere as well, but on the early Earth, it's really important to first pin down that the structure you're looking at is syngeneic and biogenic before you start interpreting it further. Another example for stromatolites is that they can have this convex laminae, and especially if this laminae thickens over the crest, because this is generally interpreted to be indicative of photosynthetic activity, so growing upwards towards the light. And another feature, this is a stromatolite in thin section, so zoomed right in at a much finer scale. You can see the stromatolite is kind of got an overhang to it. And so to maintain that angle, to maintain that structure, there would have had to have been some kind of life involved. So the EPS or the slime that binds and holds the sediment at that kind of overhang angle. All right, so now that we kind of have a background on syngeneity and biogenicity, what can rocks, biosignatures and rocks actually tell us? The main thing is the environment that they formed in, the original habitat. So I've got some examples here. These are the Strelley Pool Formation stromatolites in Western Australia. And by tying the morphology and geochemistry together, authors have been able to figure out that it was a carbonate reef environment, that looks something like this. The Tumbiana Formation stromatolites, again, by looking at the geochemistry and the morphology, they've been interpreted as a lake environment. And then these structures that have been interpreted as bubbles come from what's thought to be a hot spring system. So here we have all Archean examples of biosignatures, and they're representing distinct different environments. So the main thing that we can get is the type of environment, but we can also get other characteristics like the water depth, the paleotemperature, information on the nutrient cycling, relative sea level change over time, all this rich information. Moving on to the Turi Creek group, which is where I did my PhD work. What I'm showing here and what I'll talk about combines work from a number of different authors, colleagues mainly from the University of New South Wales in Sydney. So the Turi Creek group is a package of rocks in Western Australia. It's 2.4 billion years old, and it hosts a wealth of various different microbialites, so macroscopic biosignatures, and also microscopic biosignatures in the form of microfossils. And so we're building up over the past few years this wealth of information about this site. It's really well preserved. We know that the structures are biogenic, syngenetic and biogenic now, and it's a really good archive of an ancient environment on Earth. And so we've been delving into what these biosignatures can tell us. And firstly, just to get everyone on the same page, this is a stratigraphic column. So it basically is a representation of all of the different rock layers in the order from the oldest at the bottom to the youngest at the top. So in the field outcrop here, you can see the layering is on a bit of an angle indicated by the dashed white line, and the oldest rocks to the youngest rocks. And so that's kind of what the stratigraphic column on the left is showing you. But what the different colors don't necessarily matter for this talk. They're just representing different units and changes. But what I do want to point out is that we've been able to confirm it was a carbonate reef environment, something like this from the modern day. And a really cool thing we've also found out is we've tracked the changes in relative sea level through time. So it started off deeper, went shallow, sea level rose again, got deeper, and then it started to fall again. So all this information is recorded in the rocks and through the changes in the morphology of the stromatolites and other geochemical indicators. We've also been able to piece together a model of the depositional environment. So we think it looked something like this, with the relative position of the various microbialites and their relative water depth in comparison to one another. So it's pretty cool to be able to try and gain, build this picture of what the environment looked like 2.4 billion years ago. Another piece of information we're able to pull out, which I thought was pretty cool, this picture on the left is of the Turi Creek group, and it shows stromatolites in plan view, so looking from above, and you can see that actually elongated in one direction. And we see this in the modern day in stromatolites in Shark Bay in Western Australia, where the stromatolites are elongated because of the tide, so the current direction changes the morphology. So we've been able to pinpoint the direction of the tide billions of years ago. And then looking at the geochemistry, this gives us a heap of information about the nutrient inputs to the system, including looking at apatite, which is shown here in blue, which is a mineral, phosphorus-rich mineral, which Georgia Suarez and others have interpreted to mean that the shallow water in this system was oxygenated. So here we have this carbonate reef system with really well preserved, diverse macrofossils and microfossils, which I haven't had time to go into today, the microfossil part. But there's evidence of oxygenated environment. We've got all of these environmental kind of parameters. So we've got this kind of well-constrained system now. So part of my postdoc work, I'm leaning on this, that we have this well-constrained system. So my postdoc work now is on agnostic biosignatures. Here we've got a gradient that kind of explains the difference between, or at least my thought processes between life and non-life as we know it and as we don't know it. So in the top left, Earth-based biological processes. The top right represents Earth-based abiological processes. Bottom left is biological processes, but as we don't know it, so something different, some kind of different thing to how Earth life functions and exists. And then non-Earth abiological processes, so processes on another planetary body or another system with strange conditions that don't necessarily occur on Earth or we haven't experienced on Earth. So my postdoc research is focusing on looking at agnostic biosignatures and trying to think about what that would mean in the terms of biosignatures in rocks and especially ancient rocks. So what are universal features of life in rocks, I guess, is the overarching theme. But the issue with this is that although we have an idea of abiological processes on Earth, a sandstone is a sandstone. As far as we know, life has nothing to do with the formation of a sandstone. But do we actually know the impact of life? Life is so pervasive on Earth. We can't truly know how it interacts or influences the formation of a rock, because every example we look at through time, and especially in the modern day, Earth is life is pervaded all kind of nooks and crannies. So I guess my main question is how do we know what we're looking at and the influence of life on that structure in reference to rocks back through time? So my focus is starting from Earth-based biological processes. So biosignatures in early Earth rocks, starting from that wealth of information that we built up about biosignatures on the early Earth, and then kind of extending into the unknown towards abiological processes and trying to assess the impact of the influence between the two. So the idea is that this will help eventually branch into and looking at biosignatures from an agnostic sense, but we first need to understand the background biological processes and how they interact. So I'm using the Turi Creek group and my background in that, and all of the information that myself and colleagues have kind of uncovered about this system, and applying it to this problem. So I'm focusing on morphology and texture as biosignatures, and also elements and minerals, and thinking about how we could step back and look at these from a universal perspective, and analyzing them with a range of in situ spatially resolved techniques. This is an example here of, I've got a number of different samples, but I've just picked one to kind of, allow people to follow along. But here's an example of the stromatolite in Outcrop, what it looks like in hand sample. Then after I've cut it to make a thin section, and then the thin section slide. So this is a glass slide of the rock that we analyze and look under the microscope and other techniques. So the thing I find really interesting about this is that visually, the stromatolite is very obvious. It's the brown, bluey brown layered structure. And the sediment is also very obvious. It's the orange tan colored, mostly unlaminated side part. So visually, the life and the non-life, if you want to think of it like that, is very clear. And so my main kind of question at the moment is, what distinguishes the two? Why is the stromatolite there? Why is there no life in the sediment? I mean, what makes them different? What makes the sediment abiotic, I guess? So I've been tackling this with a number of different instruments, and the instruments have kind of been zooming down in scale and resolution. So I started off with petrographic microscopy, and although there were slight differences in the grain size and the texture, as in the patterning that you see, the mineralogy was identical at this scale, both between the stromatolite and the sediment. I used Raman spectroscopy, and this did find a difference. There was a lot of carbon or kerogen in the stromatolite and nothing in the sediment. So that was great, but also not really startling news. That's kind of expected in a fossil that of this kind of age, that there would be remnant organic material in the life part and nothing in the non-life part. So that fits with what we know. And I've also looked at this sample and a host of others under the scanning electron microscope and the electron probe microanalyzer, EPMA. And what these instruments do basically is looking at the elemental composition and the distribution of elements within the sample. And the EPMA just does that at a higher resolution. And I found that both sides of this sample, the stromatolite and the sediment, were essentially identical in the elemental composition. So the question still remains, what makes the stromatolite a stromatolite, and what makes the sediment, like what distinguishes them? Is it just the carbon? They look so different in hand sample. I don't know the answer. This is, partway through my study. The next step is to look at LA-ICP-MS, which gives an idea of the trace and the rare earth elements. So it's stepping up again in that specificity. And hopefully there will be some distinction there. We'll wait and find out. So altogether, what I hope to do with my study is to leverage the knowledge of Earth-based biosignatures from the early Earth especially, and try and branch out into these other categories, going from what we do know towards what we don't know. Thank you very much, and I look forward to any questions at the end of the session.
Great. Thanks, Erica. And as she mentioned, I think I forgot to mention this, we'll hold the questions to the end. So if you have any questions for Erica, just kind of hold on to them, and then we'll have a Q&A session at the end for everybody. And so with that, we'll be moving on to our next talk given by Joshua Krissansen-Totton, who earned his dual title PhD in Earth and Space Sciences and Astrobiology from the University of Washington. And then he moved on to do a NHFP Sagan Fellowship at UC Santa Cruz. And now he's an assistant professor at the University of Washington, where he's interested in understanding the evolution of rocky planets, both within the solar system and beyond, with a focus on questions related to what makes planets habitable and how to detect life on exoplanets. And Joshua's recent work encompasses Earth's long-term geochemical evolution, the climate of early Venus, and making testable predictions on the atmospheric evolution of highly irradiated terrestrial exoplanets. So thank you for joining us, Joshua, and go ahead.
All right. Well, thank you, James, and thank you all for being here. So I realize this the topic of this talk is a little bit broader than the kind of normal scope of this seminar series, but I really hope it'll be of interest and that the relevance to prebiotic chemistry and early Earth environments will kind of become clear. Is this advancing okay?
Yeah, everything looks good.
Okay. So I did also want to point out that a lot of the nuts and bolts calculations that I'll be presenting here were done by two really wonderful graduate students I've been working with, Maggie Thompson at UC Santa Cruz and Nick Wogan at the University of Washington. So I'll be highlighting their contributions as we go. Okay, so it really is true that the search for life on exoplanets is imminent, not only because we have these very large ground-based telescopes currently under construction, but it's possible that the first observations of inhabited exoplanet systems have actually already happened, already been made with the James Webb Space Telescope. We won't know exactly what those systems look like for a few years. It'll take a while to build up enough signal to noise, but observations of the atmospheres of potentially habitable terrestrial planets are happening. And thinking about the coevolution of life in the environment on Earth is really informing this search for life elsewhere in a direct way. So this on the left here, can you see my cursor, by the way?
Yeah, that's showing up.
So this is the Astrophysics Decadal survey on the left. This happens every 10 years in astronomy and astrophysics. The National Academies creates sort of a 10-year plan prioritizing future missions, and a very large UV optical near-infrared space telescope was the sort of recommended flagship mission coming out of that most recent decadal survey, which would be built with the explicit purpose of looking for life on exoplanets. And this figure on the right is adapted from that decadal survey. We're seeing the evolution of Earth's atmosphere here on the left, and at different times how that would appear spectrally to a space telescope. And knowledge of the early Earth is really driving the engineering requirements of these future instruments. So, for example, thinking about potentially low oxygen levels in the Mesoproterozoic and what kind of UV capabilities would be required to detect that via ozone, how that trades off with near-infrared capabilities. These are all really explicit considerations. And this figure is really remarkable in the sense that it represents decades of geologists and geochemists thinking about the ways in which life has modified the environment on Earth, and then taking that knowledge and using it to kind of build these machines to peer upwards. I think it's a really inspiring vision. Now, if you've heard anything about exoplanet biosignatures, I'm sure you've heard that oxygen is potentially a promising sign of life, a product of oxygenic photosynthesis. But of course, what we know from Earth history, and this is a figure from Catling and Zarnley review paper Earth's atmospheric composition from 4 billion years ago to present, we know that oxygen levels in the atmosphere have only been readily detectable during the Phanerozoic, perhaps detectable at times in the Proterozoic, depending on exactly what proxy estimates you tend to believe. But for about half of Earth's history, there was certainly no free oxygen in the atmosphere. And so it's reasonable to ask, how might we go about detecting life on these sorts of exoplanets where there is life present, but anoxic atmospheres? And one thing that we do think we know about the Archean atmosphere is that there was likely very abundant atmospheric methane that was almost exclusively biogenic. And the various lines of evidence for this, originally, this sort of comes from thinking about anoxic analog environments and scaling them up in global ecosystem models. Of course, atmospheric methane provides a mechanism for Earth's, the oxidation of Earth's crust, but there's also more recent evidence, independent evidence for this high methane in the form of xenoisotopes, and you can check out this review paper to hear more about that. So what I want to make the case for today is look at methane as kind of an interesting case study of potentially useful exoplanet biosignature for Archean analog exoplanets with anoxic atmospheres. And so this is a three-part argument, a three-part talk. I'll firstly try and convince you that if life is common in the universe, then methane-producing biospheres are potentially quite common. This is really an argument about evolution. The second part is about non-biological processes. How might we be fooled into thinking we've found a methane biosignature through non-biological mechanisms? And I'll also try and convince you that this is something worth thinking about because methane is imminently detectable with current and near-term telescopes. All right, so part one, if life is ubiquitous, then methane-producing biospheres are probably common. So there are a number of kind of dominant pathways by which methane is produced biologically on Earth today: hydrogenotropic methanogenesis and carbon monoxide oxidation. And what is important to note here is that the substrates needed are likely to be abundant on terrestrial planets in general, and that CO2 and water will be present on habitable planets by definition, and then the other substrates needed, hydrogen and CO, are likely to be fairly common outgassing products. And so methanogenesis, in many ways, is a logical evolutionary response to sources of environmental free energy, the geochemistry present in the environment. And so we might expect life elsewhere, even if its biochemistry is radically different, to exploit these same sources of free energy. Now, additionally, methane on Earth is produced by acetoclastic methanogens, essentially the decay of organic matter. And so this adds an additional boost of methane for any life that's based on organic carbon. Additionally, there's various lines of evidence to suggest that methanogenesis emerged very early in Earth's evolution. Phylogenetic reconstructions and reconstructions of the last universal common ancestor all seem to indicate an origin for methanogens that is essentially coincident with the origin of life within era. And so this is a metabolism that has seemingly emerged very early in response to readily available environmental free energy. And so it's plausible to expect similar metabolisms to emerge in terrestrial planets elsewhere. And it's something we also see in models of Archean ecosystems and other anoxic models of anaerobic ecosystems, is that for a range of plausible chemoautotrophic metabolisms, methanogens, acetogens, acetotrophs, etc., you find that very methane-rich atmospheres are a common outcome of these sorts of biospheres across these different ecosystem models. And finally on this, I don't want to spend too much time on the details here, but this is something we discovered when we first started thinking about methane in the Archean. The chemical equilibrium calculations showing that methane was out of thermodynamic equilibrium with the Archean atmosphere ocean system due to its replenishment by life. And this is a theme, or related to a theme we'll keep coming back to, is that methane and carbon dioxide represent carbon at opposite ends of the redox spectrum, opposite levels of oxidation. And so what we'll find is that it's difficult for non-biological equilibrium processes to produce both atmospheres rich in both methane and carbon dioxide without producing a whole lot of intermediate carbon monoxide. All right, so having hopefully convinced you that methane-rich atmospheres is not a surprising product of biology, I now want to turn attention to how you might be fooled into thinking with found life, so non-biological mechanisms for methane production. And this is a topic that has been thought about for a while, at least since the 90s. There are many papers that mention methane biosignatures in passing as a possibility, but with the careful caveat that there are non-biological processes that can also produce methane. And so we wanted to try and be a little systematic here in going through these different plausible mechanisms to figure out, well, exactly how much methane could you produce without life. So we'll talk about some of those sort of most obvious false positives today: outgassing impacts, low temperature systems, and very volatile rich planets. Now, what is definitely true is that methane is going to have a very short lifetime in the atmospheres of terrestrial planets. And this is true regardless of atmospheric bulk composition. It's true in oxidizing atmospheres, as outlined here in the summary of the chemistry. Methane is very rapidly destroyed by reaction with OH radicals, but even in more reducing atmospheres, methane is still photodissociated. The hydrogen produced by that is irreversibly lost to space, and you may build sort of larger hydrocarbons with what is left over, but essentially that methane is not coming back. And this can be quantified as Maggie did in a photochemical model here, plotting the different CO2 mixing ratios and the different steady state methane mixing ratios. What is the flux of methane you need to sustain that much methane in the atmosphere? And indeed, you do find that for part per thousand levels of methane, you need biological fluxes of methane to sustain this. And this is consistent with many other photochemical calculations in anoxic atmospheres. So the key question for us really becomes then, could you produce large fluxes of methane into a terrestrial planet atmosphere without life? And a useful starting point for this discussion is the modern Earth's methane budget. This figure is from the recent IPCC AR6 report showing sources and sinks in Earth's atmosphere today. And of course, biology is the dominant source, as are anthropogenic sources, which are biology by another name. We do see geological sources labeled here on the left, which is small but not insignificant compared to these biological sources. But what is important to note here is that what is termed geological sources is really predominantly indirect biology. So a lot of this is thermogenic. Methane produced by the decomposition of organic matter that was previously living biomass, mud volcanoes. The amount of methane here that is genuinely non-biological is it's hard to quantify, but it's certainly very small. The reason why it's hard to quantify is that life has so thoroughly modified the composition of Earth's crust over geologic time that it's you almost have to run a whole counterfactual experiment to get at this number. Well, what would the methane flux be without life? It's something that's very difficult to know. And so our approach, rather than to try and do this very difficult decomposition, was to do some first principles calculations for particular mechanisms to see how they could plausibly scale up on other planets. And so that's something that Nick Wogan did a few years back. Now he built a model of magmatic outgassing, the exclusion of volatiles from partial melts, and considered a very broad range of planetary conditions, so a wide range of redox states of melt volatile composition, temperature pressure conditions, to calculate how much methane you could plausibly get out of these systems on exoplanets of arbitrary composition. And in general, what Nick found is that it's difficult to make a lot of methane. Methane fluxes are typically, from these magmatic systems, are typically orders of magnitude less than biology. And in the rare instances where you do produce appreciable methane from these systems, it tends to be accompanied by a lot more carbon monoxide, as I alluded to previously. And there are a number of reasons for this, why you don't get much methane out of volcanoes. Firstly, water is very soluble in partial melts, so it's hard to get a lot of hydrogen atoms into the gas phase. Secondly, typical partial melt temperatures are just too hot for methane to be thermodynamically favored. And finally, you need very low oxygen fugacities to make a lot of methane, which tends to also produce a lot of carbon monoxide. So that's very quickly the take home from those calculations. The result here is very different to what you'd expect on an inhabited planet, where any carbon monoxide that is produced is going to be rapidly consumed. This CO oxidation metabolism is something that's, we think, comparatively easy to do. If there's water around, this is a metabolism that has seemingly evolved independently several times on Earth with different metal active sites, and is very widespread across the microbial genome. So on inhabited planets, you would not necessarily expect a lot of carbon monoxide along with the methane. And this figure is kind of an illustration of that conjecture. We're plotting here the atmospheric CO to methane ratio as a function of the gassing flux from the interior, and then everything plotted different, down the bottom here are different ecosystem models with different combinations of chemoautotrophic metabolisms, whereas the blue data points up here are those same models without life, so lifeless planets. And you do indeed find that once there is life around, it does tend to draw down the carbon monoxide and produce low CO to methane ratio. So this is a very crude illustration. The details are, of course, going to depend on the cell spectrum and so on, but we think that CO to methane ratios might be a useful diagnostic for disentangling these different sources of methane. I won't say too much about impacts as a false positive, other than to say that a similar story applies, and that it's hard to make methane and CO2 without carbon monoxide. But probably the most the most well-known potential false positive for a methane biosignature is methane produced in low-temperature systems, water rock reactions, serpentinizing systems. And so this is most often thought about in terms of like a water rock reaction producing hydrogen, and then this hydrogen reacting with CO2 by Fischer-Tropsch type reactions to produce methane. But this isn't the only way you could make methane. There's also direct oxidation of siderite, as well as reactions of carbonate with hydrogen that could in principle produce methane. But what is critical to all these mechanisms is that if you're in an environment sufficiently oxidizing to have the CO2-rich atmosphere, then you need to explain where the reducing power is coming from to continually produce this methane. And that typically comes down to what is the supply of iron to the surface. And of course, this is a complicated problem depending on tectonic setting and geochemistry on many temporal and spatial scales. But we can at least try and make some crude estimates of what is plausible in terms of methane production from these systems. And this is a figure from the first attempt to do that back in 2018, where we said that the maximum amount of methane you can make from these water rock reactions is ultimately limited by the rate at which you're delivering iron and newly produced crust to the surface, how much of that is actually getting oxidized to make hydrogen, and how much of that is actually being converted to methane, and even making very generous assumptions about how efficient the different steps in this process are. You end up with a distribution for how much methane you can make non-biologically that is considerably less than biological fluxes. And Maggie has recently revisited this so that shaded pink distribution is the same as the blue one here. It's just in log space now. And Maggie went through the literature looking at different estimates of sort of maximum methane fluxes from low temperature geochemical systems, serpentinizing systems at mid-ocean ridges, subduction zones, and generally found these estimates to be consistent with the kind of ballpark max flux calculations that we did previously, all of which are considerably less than biological flux of methane. You might be wondering about sort of Titan-like exoplanets. Again, I won't spend too much time on this. Happy to chat about it in questions, but the take-home point here is that while there is methane in Titan's atmosphere that's being replenished from the interior, if you take something Titan-like and put it at Earth-like separations and melt the ice, the lifetime of that methane is going to be very short compared to geologic timescales. So what all of this is hopefully working towards is an acknowledgement that, yes, there are these processes that can make methane without life, but we think that, at least for these sort of most obvious false positive mechanisms that we've talked about, it might be possible to make contextual observations of the planet, look at the other gases in the atmosphere and so on, to deduce whether this is likely a false positive or something that's more consistent with microbial production. And then the final thing I wanted to mention, why this is worth thinking about, I wanted to say something about the detectability of methane with current and future instruments. These are some calculations I did a few years ago, just simulations of what the James Webb Space Telescope would see looking at a potentially habitable nearby exoplanet. And it does indeed seem like methane detections should be possible, and you don't have to take my word for this. There've been a number of subsequent studies that have done similar calculations, have come to similar conclusions that methane is among the most detectable of atmospheric gases. In fact, it's so detectable that there have already been claims of methane detections in rocky planet atmospheres using Hubble. This is a hot planet that isn't habitable, and as it turns out, this detection has been very strongly contested, but the fact that it's sort of at the limits of what Hubble can do very much suggests that this is in the realm of possibility for the James Webb Space Telescope. And it's also worth mentioning you don't just get a detection of methane. It may be possible to get constraints on the atmospheric abundances of these different carbon-bearing gases, which could allow us to at least tentatively rule out some of these most obvious false positive scenarios that we've discussed. And in fact, detecting Archean Earth levels of biological methane with JWST is probably going to be much easier than detecting modern Earth levels of oxygen or ozone with JWST, just due to the different wavelength ranges required. Finally, though, I did want to point out that we're not going to get anything definitive with just the James Webb Space Telescope. There's a lot of information about the planetary context that will not be accessible to us. Things like the presence of water vapor will not be something that can be measured. So definitive life detection will have to wait for future instruments. But that is the case of methane as a biosignature in a nutshell. Just to add a bit of, or to finish with a bit of caution here, we've really only discussed the most obvious kinds of methane false positives, and of course the parameter space for terrestrial planet composition, terrestrial planet environments is potentially vast, and there are a lot of unknowns in what processes could generate a lot of methane. And so I've listed some sort of interesting open questions here about whether it's plausible to have sufficient mineral catalysts in low temperature habitable environments to produce a lot of methane abiotically. Are there other sort of more exotic reactions making methane that are not significant on Earth that could be scaled up on exoplanets? So what will ultimately be needed here are models that explicitly couple planetary interiors and crusts and atmospheres to explore some of these more exotic false positive mechanisms. But what I've hopefully convinced you of is that, more broadly, understanding the coevolution of life in the environment on Earth can be directly used in the search for life elsewhere. And I've talked about methane-rich Archean analogs as one potential example for that. At least I find it compelling because it's difficult to sustain high methane abundances without very large fluxes, and it's difficult to produce large fluxes of methane abiotically, especially in combination with low carbon monoxide and a lot of CO2. And this is worth thinking about because methane is one of the most easily detectable atmospheric constituents for terrestrial planet atmospheres with the James Webb Space Telescope. And I'm really excited to hear your comments and questions, because I think this community, people thinking about prebiotic chemistry in early Earth environments, could have a lot of great ideas about how we might be fooled into thinking we've found life, ways we could generate a lot of methane. So with that, thank you again, and look forward to the discussion.
Great, thanks Joshua, and thank you also to Erica and Don. And with that, we've gone through all the talks, and so now the chat should be open for questions, and you can also raise your hand, and using the feature in Zoom, it will ask you to unmute. And so if you have questions for any of the speakers, go ahead and get them in. And I also already see, let's I guess ask Kevin Devine. I think you should be able to unmute yourself and ask your question.
Yeah, okay. So thank you, Erica and Joshua, for two very good presentations, which I really enjoyed. So I got a question for both of you. First of all, Erica, you're looking at your combinations of stromatolites and sediments. I'm interested in your carbon-rich molecules. I would guess, I imagine they're a mixture of everything from polyaromatic hydrocarbons to carbonates. Have you looked at the carbon-12, carbon-13 ratios in these organics? And if so, are they suggestive that they're biogenic in origin? And likewise, is there any sign of any organic material in the sediments?
Hi, thanks for your question. My focus for my PhD was actually on microfossils within this reef environment. So a preliminary study I did as part of an honours project, which is a one-year research program in Australia, was on the stromatolites, and then I zoomed in and looked at the microfossils. So I have done in situ carbon isotopic work on the microfossils, but not carbon isotopic work of carbon isotopes on the stromatolites, because the microfossils were actually my focus, and as I mentioned, I didn't have enough time to delve into those. But yes, those results are indicative of biology in the minus 20 to minus 36 range. They vary from.
Okay, yeah, indeed, very good. So that's, yeah, thank you very much. And a question for Joshua. I see Alex Dean Dro's almost read my mind. I was going to ask you about the methane fluxes on Mars that we're seeing. You know, it's a great shame the tunable laser mass spectrometer couldn't look at the isotope ratios for these methane fluxes, because they appear to be seasonal, rather like they are with biological processes on Earth. But I think the carbon monoxide to methane ratio on Mars is high. So would that, I mean, it may just be that we're looking at a very low level microbial ecosystem, if any. Just want to know what's your thoughts on that?
Sure. So I guess the first thing to mention is that the methane detection claims on Mars, those abundances that have been reported are, orders of magnitude less than what I was sort of presenting.
Oh yeah, they are. They're parts per billion. Yes, indeed. But,
right,
but
right.
they do fluctuate seasonally. Yes.
Yeah. So I guess the other thing there is we actually have this other paper. Stephen Scholes was the first author on that, looking at the carbon monoxide question on Mars, as you discussed, and trying to put some limits on that, saying, well, if there was life on the subsurface today, and it was consuming that CO, how much biomass could you have and still have consistency with the observed atmosphere? And so we put some rough constraints on that. And indeed, you can't have a huge amount of biomass, perhaps unsurprisingly. So that is a calculation that can be applied to Mars.
Okay. Thank you very much. Once again, two very good presentations, which I found fascinating. Thank you.
All right, so we do have some questions in the chat, and the first one is from Uli Muller, and sorry, I scrolled off of it, but it's to Erica, and I guess it says, it may lead to incorrect conclusions to compare the biogenic stromatolite with the sediment immediately beside it, because the sediment near the stromatolite can contain leaking cells and or metabolic products of the biological structure. Would it be good to include a control from the same age, but physically distant from the organisms?
Thanks for your question. And yes, 100%. And I have got that as part of my greater study. I just picked a very, I guess, really easy to understand sample here because it showed the sediment and the stromatolite together. But I have got a sample suite that's got definitively biological, so stromatolites and other microbialites. As far as we can tell, definitively abiological, so just plain sediments from the same system, but as you said, further away stratigraphically, so they're not at least directly related to the microbial features. And then a third category, which is mixed, so flat microbial mats, where they've got in essence, the microbe layers and then the sediment, and it's interlayered. So I've got these three categories that I can hopefully tease apart what's going on, but yes, I definitely have more of a control side of things. Thanks for your comment.
Great. And I think Uli had a second question for Joshua. And the question is, the calculations of methane production seem to be using global averages, which is useful for detection in atmospheres. But to what extent could methane and carbon monoxide or hydrogen have existed in much higher local concentrations? And this is important for thinking about plausible environments for origins of life.
Sure, yeah, I mean, that's absolutely true. I think in the exoplanet context, we tend to think more about global averages for a couple of reasons. Firstly, that's all you're going to get in the near term from these exoplanet observations, is some integrated measurement. But I think it's also true that methane is probably going to be, and these other carbon-bearing gases are going to be pretty well mixed in the atmosphere. There might be, some spatial variation, but that kind of well-mixed assumption is probably fairly reasonable. But yeah, certainly if you're asking questions about prebiotic chemistry in local environments, then a local scale is going to be relevant.
So I guess there was another question from Thomas Klein, and he asks, I wonder how the frequency of encountering high methane atmospheres would affect your confidence in a biotic origin. I would think rarity might be the most convincing.
I think that's true. Yeah. I mean, if we were in a situation we found methane that was hard to explain by any biological mechanism we knew of, but we kept finding planets that looked like that, then you would be suspicious that there was something you're missing, some non-biological mechanism that was explaining the multiple planets. And so I think in the end, methane alone is not going to be an unambiguous biosignature, right? We're going to need other corroborating signs of life in addition to the methane to really understand what we're looking at.
Great. And so I believe that Alex Dean's row question got kind of answered when Kevin asked his question. So I see another question in the chat from Preetha Jaipal, and that's to Erica. And it's asked, what would you say are the more useful or effective methods to detect the syngeneity of stromatolites in the deposits, as well as other biosignatures with their environment?
Thanks for your question. I guess if you had to kind of look at it from the most useful or most effective way to go about addressing syngeneity and biogenicity would be to start, at least in my opinion, to start from the fields scale. So be there in person, see the field relationships, see what the layers are doing, see the structure in, and how it corresponds with the sediment in the field setting, and mapping the various units so you can really get a feel for the system. And then going from there and I guess zooming closer and closer and closer. So looking at it under the microscope, doing various analytical techniques, and getting information from a wide array of scales, and that can really help pinpoint collectively biogenicity and syngeneity, because there's not generally considered not to be one smoking gun as such that says yes, this is biological. There's always people questioning and querying what's going on. So the more lines of evidence you have from the multiple scales, the better and stronger your cases.
Great. And I saw that there were a couple comments just talking about methane on Mars. There, people were just providing some more information in the chat. So if you were interested in methane on Mars, I think you could take a look at the comments from David and Don. Let me see. There was, I guess, a question from Anandita Mandal for Joshua. Apart from methane, can we use rubisco as a biosignature molecule?
I wonder if they could elaborate on that question. I'm just not quite sure what.
Yeah, I guess if you wanted
for
to
that.
clarify, you could put it in the chat. But if not, we could. And then I think I saw also that maybe Deepan had a question, but I think Don responded in the chat to that question. So I think as of now, we might have gone through the questions. I don't see any raised hands. So maybe I'll just quickly wait for a little. I'll say something, and if you have a question that pops up, go ahead and bring it up. But I guess I'd remind everyone that in a couple weeks, there will be the Pc3 Workshop that Zach mentioned at the beginning. So please read up about that and join if you can. And also related to that, the seminar series, we won't have another seminar 3 weeks from now because we don't want to have too many Pc3 things going on right close together. So we'll resume the seminar series 6 weeks from now. And so I don't see anything. Actually, I saw that I just saw that Kevin has his hand raised. So let me see. I can ask you to unmute. if you have something to say, Kevin.
Rubisco, it's the enzyme that's principally responsible for carbon fixation. It's the most abundant enzyme on this planet, and it's present in every leaf of every plant. So, and I think you can maybe detect it spectroscopically. I'm sure it can be detected by satellites above the Earth. I'm not sure if it could be detected across sort of interstellar distances. I'm not sure about that. But that's what Rubisco is. Certainly a biosignature, it certainly would be.
Just to add to that, yes, I don't think that's something that is sort of within the realm of interstellar detection, but perhaps the carbon fractionation that it imparts is something that could be additional evidence that could be leveraged to interpret methane in a planet's atmosphere. I mean, that would be a very challenging measurement, but if you could get carbon isotopes for methane and CO2, then that could potentially discriminate from a number of non-biological processes.
Okay, yeah, so I saw that I'm glad we took some time because another question popped up in the chat, I think for Erica from David Catling. And regarding stromatolite versus sediment, it looks like question 1 is, isn't the conclusion that elemental composition insufficient unless you're looking at reduced carbon? And I guess question 2 in that regard is a black shale, a biosignature of photosynthesis. I'm not sure it could be produced otherwise.
Hi, David. Thanks for your questions. I'm not quite sure what you mean by question 1. Isn't the conclusion that elemental composition is insufficient unless you're looking at reduced carbon? Sorry, I'm not quite sure what you mean. If you could clarify, I can try and. Oh.
Yeah, maybe if you raise your hand, David, I could unmute you, or I could let me look for you on the list. Okay, I think you should be able to unmute, David.
Well, can you hear me?
Yes.
Okay. Well, I thought you said that basically the elemental composition was identical between the sediment and the stromatolite apart from the organic carbon. So that's the basis of my question, that it just doesn't seem like the elemental composition on its own is informative unless you're specifically looking at total organic carbon. And then that leads on to the question, well, if there's so much organic carbon that you've got a black shale, is that only the case of photosynthesis, which I think it is on Earth, but maybe there's a gray area?
I'm not sure about the shale question. Maybe Dawn can weigh in on that. As regards to the first question, yes, I mean, there's looking at the when I said the elements were the same, I wasn't talking about quantitatively the same yet. I do have that information, but I literally collected it last week, so I haven't had a chance to look at it from a quantitative angle, but a qualitative angle, as in this element exists in both SEM maps, which is non-quantitative, in quite a high distribution, a higher abundance in a non-quantitative sense, because it's appearing in the maps. That was more what I was talking about. Does that kind of address your first question, or did I miss something further?
Sorry, I couldn't unmute again. I got disallowed, forbidden. Yes, I think it does. So you're talking more about the distribution rather than the quantities. Is that what you're getting at?
Yeah, in this talk, it was, sorry if that wasn't clear, it was more about the presence and distribution, and the quantitative info is coming. Yeah.
Okay.
Great. Sorry.
Anything to add on the shale and photosynthesis angle?
So I guess there's a little bit of a question about, like, a black shale implies high organic matter, because you can have black shale, you can have shales that are reduced without high organic matter as well. And so I think, you know, the question about whether you can get an organic rich rock without the presence of life really depends on sort of the abiotic production of organic matter, which is, we know it happens because you have to have it for the origin of life, and we see the organic matter in interstellar materials, and it's on Mars, but we don't know whether it's from life on Mars or not. And so, sort of like, what is a biosignature in terms of organic matter is something that intuitively I feel like photosynthesis is taking light energy and turning it into chemical energy in the form of organic. So that's the best way to get a lot of organic matter. But we don't understand how organic matter behaves on an abiotic planet surface. And so, and we don't know, like there's most of the organic matter that's produced gets degraded by microbial communities on Earth. So you also have this question about whether you can produce abiotically a whole bunch of organic matter and then not have it decay. Maybe you could get a high concentration. Intuitively, I say I don't think that's the case, and I like photosynthesis to make organic rich rocks, but I have to admit that I can't prove that.
Just to add a quick comment to that, the answer to that question is also really crucial for how much methane you can make non-biologically, right? If there are mechanisms by which you can generate massive amounts of organic matter without life, then you can imagine that translating to pretty large non-biological methane fluxes.
Okay, great. I think that kind of brings us to the end of the questions that I saw in the chat. And I did see that there were some kind of conversations going on between people in the chat. So I just quickly posted the link for the PC3 Slack channel. Everyone's free to join, and it's not that active, but it's there. So if you guys want to continue some of these chats there at any time, you're free to do that. And I guess with that, we're kind of nearing the end of the time slot. So I guess we can go ahead and thank all the speakers again for their great talks and discussions. So yeah, thank you. And we hope to see some of you at the PC3 workshop in a couple weeks and the next PC3 seminar in about 6 weeks. So thank you everybody for joining us. And thank you to the speakers.
Thanks.
Great. Bye, everybody.